Welcome everyone. Today we will continue our lecture on Enterobacteriaceae, focusing on Escherichia coli, Klebsiella, Salmonella, and Shigella. And in this lecture we will study their important characteristic, disease, and laboratory diagnosis. E. coli, Klebsiella, Salmonella, Shigella are medically important enteric gram-negative fruit, belongs to Enterobacteriaceae family. These organisms are important in medical laboratory, as it commonly causes intestinal infection, urinary tract infection, septicemia, and hospital-acquired infection. As I mentioned in a previous lecture, the identification begins by lactose fermentation on Makongi agar. Lactose fermenter organism produce pink colony, and the non-lactose fermenter produce colorless colony. The lactose fermenter bacteria like E. coli and Klebsiella, we can differentiate between them by motility test, as E. coli is motile while Klebsiella is non-motile, and the non-lactose fermenter like Salmonella, Shigella, and Proteus. Salmonella and Proteus are motile bacteria, whilst Shigella is non-motile. And we can differentiate between Salmonella and Shigella, as Salmonella produce hydrogen sulfide while Shigella don't produce hydrogen sulfide. And Proteus produce the urease enzyme, is positive for urease enzyme. This classification is important for identification of Enterobacteriaceae. And one of the most important members of the Enterobacteriaceae is Escherichia coli or E. coli. It was first described by Theodor in 1885. It's a gram-negative, that is mean on a gram stain under the microscope it appears red. It is root bacteria, bacilli shape, facultative anaerobic. It can live in oxygen and without oxygen. As I mentioned, most bacteria that are cause disease is facultative anaerobic and usually motile. Some may have the capsule. And this bacteria is normally lives in intestine as a part of a normal flora. However, some strains become pathogenic and can cause disease. It may also found in a soil water contaminated with a fecal material. For example, if E. coli enter the urinary tract, especially in female, it may cause the urinary tract infection. Although the E. coli normally lives in intestinal, some strains become dangerous or pathogenic because they have the special virulence factor. Virulence factor of pathogenic E. coli. Pathogenic strains of E. coli have several virulence factors that help them to attachment, invasion, and disease production. And these virulence factors may include pili or fembri that help bacteria to attach to body tissue. For example, uropathogenic E. coli use the pili to attach the urinary tract and cause UTI. Flagella that help bacteria for movement. Capsule protect the bacteria from phagocytosis. The hemolysin, these toxins damage the host cell. And other virulence factors like exotoxin, as some strains produce the enterotoxin that cause diarrhea, and endotoxin, the lipopolysaccharide in a cell wall, may produce fever and inflammation. And because of these virulence factors, E. coli can produce many different diseases. And the most important point, the difference between the harmful intestinal E. coli and pathogenic E. coli is the presence of these virulence factors. Disease caused by E. coli. E. coli can cause both intestinal and extra-intestinal infection. That means it can cause infection in the intestine and other parts of body. And the most common disease is urinary tract infection, especially by the uropathogenic E. coli strain. For example, patient with burning during urination and positive urine culture may have UTI caused by E. coli. Other infections may include septicemia, wound infection, soft tissue infection, hospital-acquired pneumonia, neonatal meningitis, diarrheal disease. And the most dangerous strain is E. coli O157H7. This dangerous strain that causes severe bloody diarrhea. And after understand the disease that caused by E. coli, we should understand how to identify E. coli in lab. And the laboratory diagnosis of the E. coli began with a specimen collection. And the most possible specimens are urine, stool, blood, pus, body fluid, it according to the site of the infection. For example, if they are UTI infection, we collect urine. If they are bacteremia, we collect blood and so on. On a gram stain, when we make the smear, the E. coli appear as a gram-negative root. That is mean. When we done the gram stain, it take the red color because it's a gram-negative bacteria and it's bacilli in shape under the microscope. When we culture on Makongi agar, it form pink colony. Why? Because it ferment the lactose. And on azomethylene blue agar, it produce black colony with a metallic green shine. Why this color? Because E. coli strongly ferment lactose and produce large amount of acidity. After gram stain and culture, we make the biochemical reaction. And the most important biochemical reaction for identification of the Enterobacteriaceae is INVIC test. When we done INVIC test, E. coli is positive for indole and methylred, while negative for Voges proscauer and citrate. And it can ferment glucose and lactose, and it's motile bacteria usually, non-forming spore, and don't produce hydrogen sulfide. Now we will move to another important bacteria in Enterobacteriaceae family, Klebsiella. Klebsiella pneumoniae are the most important species of the Klebsiella. It's a gram-negative root, non-motile, facultative anaerobic, and covered by a thick capsule. And it's present normally in intestine but may also survive in a moist hospital environment, making them the most important hospital-acquired bacteria as, for example, Klebsiella can spread in hospital through the contaminated equipment or the hand of healthcare worker. And unlike E. coli, Klebsiella has a prominent polysaccharide capsule. This capsule makes them more virulent and give colony mucoid appearance. It has a polysaccharide capsule. And morphologically, it typically shorter and thicker than other Enterobacteriaceae family. Pathogenicity of Klebsiella. As I mentioned, the polysaccharide capsule is the most important virulence factor that help the Klebsiella to make a disease. It protects the bacteria from the phagocytosis, and because of a capsule, the colony are large and mucoid. Klebsiella pneumoniae are commonly cause pneumonia, urinary tract infection, septicemia, wound infection, also important cause of a hospital-acquired infection, especially in immunocompromised patients. For example, a diabetic patient or patient in the ICU may develop pneumonia. Therefore, we should understand how to identify Klebsiella in the lab. And laboratory diagnosis of the Klebsiella, we begin with a specimen collection. As I mentioned, it depends on the site of the infection. In case of the pneumonia, as it commonly causes pneumonia, we collect sputum. In case of the UTI, we collect urine. In case of wound infection, we collect pus. In case of the meningitis, we collect CSF and so on. After that, the first step in identification is a smear. Make the smear on a gram stain. It's a gram-negative fruit. It means it appears red and bacilli under the microscope, but this is non-specific, as all Enterobacteriaceae family are gram-negative fruit bacteria. Culture on Makongi agar, Klebsiella produce a pink colony because it's lactose fermenter bacteria, but the colony of the Klebsiella is a large and mucoid due to the capsule, the thick capsule. And when we done the biochemical test, INVIC test, unlike E. coli, it's negative for indole and methyl red, positive for Hojas proscauer and citrate. And many strains produce beta-lactamase, making them resistant to the ampicillin and amoxicillin. Severe infection may require cephalosporin. As I mentioned, it's a gram-negative root bacteria, and Klebsiella is usually thicker and shorter than other Enterobacteriaceae. And on Makongi agar is produce pink colony, but the colony of the Klebsiella, unlike E. coli, is a large and mucoid. E. coli produce a dark pink small colony, while Klebsiella produce a large mucoid colony. Both of them is lactose fermenter and both produce pink colony, but one, like E. coli, produce dry colony, while Klebsiella produce large mucoid colony due of a capsule. The capsule is a special characteristic of the Klebsiella. Now we move to another important bacteria in Enterobacteriaceae family, Salmonella. Another important member of Enterobacteriaceae is Salmonella, which is considered as a pathogenic bacteria. Salmonella and Shigella is not part of a normal flora. It's considered pathogenic bacteria. It's pathogenic for human and animal, and it's commonly affect the human through contaminated food or animal product. For example, undercooking chicken, eggs, or contaminated milk may transmit salmonella, where they cause enteritis, inflammation of the intestine, usually causing diarrhea, systematic infection when bacteria spread through the blood and affect the whole body, like in septicemia. Enteric fever is a specific type of infection, usually it's caused by Salmonella typhi that leads to high fever, abdominal pain, or rash. And the characteristic of Salmonella is like Enterobacteriaceae family is a gram-negative root bacteria, non-spore forming, facultative anaerobic, and motile. And Salmonella can ferment the sugar like glucose, mannitol, maltose, forming acid and gas, except Salmonella typhi that produced acid but no gas. And there are several classifications to classify the Salmonella according to the clinical classification, antigenetic classification, and molecular classification. And I will go to explain this classification in the next slide. Clinically, we can divide Salmonella in two groups: typhoidal Salmonella and non-typhoidal Salmonella. Typhoidal Salmonella that cause enteric fever, and non-typhoidal Salmonella that are usually cause the gastroenteritis and food poisoning. Typhoidal Salmonella include Salmonella typhi and paratyphi, and these only infect human and cause enteric fever. This come only from humans. Infection happen when injection food or water contaminated with a feces from any infected patient, while non-typhoidal is found in animals that cause food poisoning or gastroenteritis. It happens when eat contaminated food such as undercooking chicken, eggs. They can acquire non-typhoidal salmonella, and that's usually cause diarrhea, abdominal pain, sometimes vomiting. These bacteria, non-typhoidal, come from animals or animal products, not from other humans. In addition to the clinical classification, we can classify Salmonella according to the antigen, as Salmonella has two important antigens. O antigen, somatic antigen. Why it's called somatic? Because it's part of a cell wall lipopolysaccharide, and it's used in a Weddell test. When the O antigen reacts with O antibody, the granular clump appears. Serology test. Antigen-antibody reaction that lead to the granular clumping appear. And H antigen, this is present on a flagella that give the organism motility. The O antigen help in a serotype classification, serogrouping, while the H antigen help in identify motile salmonella. Typhoid salmonella pathogenesis, it means the process of the infection, how it happened. Typhoid fever or enteric fever, it caused mostly by Salmonella typhi. The bacteria enter the body by contaminated food or water, oral route. Infection dose is higher than Shigella. What we mean here? It means more number of the bacteria required to cause the infection than Shigella. Shigella, small number of the bacteria can cause the infection, while in Salmonella, large number of the bacteria required to cause the infection. After we get Salmonella by contaminated food or water, they enter the intestinal epithelial cell, then they cross the submucosa, and they are taken up by macrophage. They spread to bloodstream, reach liver, spleen, lymph node, and bone marrow, may reach gallbladder, kidney, lung, multiply in the intestinal lymphoid tissue, and excreted in the stool. The incubation time is a time between getting infection and showing the first symptoms is usually 10 to 40 days, and the symptoms include headache, chills, gradual fever, and sweating. For example, a patient with a prolonged fever after drinking contaminated water may have typhoid fever. The laboratory diagnosis, it includes blood culture in the early stage, especially for typhoid fever, because bacteria are present in the bloodstream during the first week, whereas stool and sometimes urine culture are most useful later when the organism excreted from intestine. On a gram stain is gram-negative root bacteria, but this is non-specific. On Makongi agar, they produce colorless colony because they don't ferment lactose, while in Salmonella Shigella agar produce black colony center due to H2S. They produce the hydrogen sulfide, and most Salmonella species on MVEC test is showing indole negative, while methylrate positive, Voges-Proskauer negative, citrate positive. And Weddell test it only used for typhoid fever that caused by Salmonella typhi. A serological test, antigen-antibody reaction, and the positive result appear as a clumping, while the negative they are non-clumping form. Now, the non-typhoidal salmonella, how it's acquired? It acquired from animal and animal product. It includes all salmonella species except Salmonella typhi and paratyphi that affect only human. While the non-typhoidal salmonella, it's acquired from the animal and animal product after eating contaminated animal products such as undercooking chicken, eggs, meat, and other, and it is common in hospital. The infection happens after enter the intestine, and the organism produces a strong inflammatory response with marked neutrophil infiltration of the intestinal mucosa that lead to gastroenteritis, the inflammation of the gastrointestinal tract, and the symptoms of gastroenteritis characterized by nausea, vomiting, watery diarrhea, fever, abdominal cramps, and about 8% of patients may develop bacteremia, the presence of the bacteria in blood, which can then reach to the heart, blood vessels, bone, or other organ. At the end, we concluded that this large group of the gram-negative root bacteria, Enterobacteriaceae family, are medically important as it can cause many diseases like UTI infection, pneumonia, enteritis, and others. All these membranes are facultative anaerobic and motile except Shigella and Klebsiella are non-motile bacteria, and some of them ferment the lactose like E. coli, Klebsiella, and produce pink colony on Makongi agar, while other Salmonella, Shigella, Proteus are non-lactose fermenter and produce colorless colony on Makongi agar. And the identification of this large group we can identify by the colony morphology and biochemical tests like the INVIC test.